everyone. My name is Hanan Akila binti Kamar Zaman and I'm from SMK Padang Tembak. Today, I will be explaining an exciting scientific technology called CRISPR. CRISPR stands for Clustered, Regularly Interspaced Short Palindromic Repeats. Even though the name sounds complicated, the idea behind it is simple but powerful. CRISPR is a gene editing technology that allows scientists to change DNA inside living organisms with high precision. This technology is transforming fields like medicine, agriculture, and biological research, and it has the potential to solve major global problems such as genetic disease and food shortage. In this video, I will explain what CRISPR is, how it works, its applications, the challenges involved, and its future potential. Before we begin, my topic today connected to two Sustainable Development Goals, which SDG 3, Good Health and Well-being, and SDG 2, Zero Hunger. So first, what exactly is CRISPR? CRISPR is a tool that scientists use to edit genes, which means they can add, remove, or change parts of DNA. DNA is the molecule that contains the genetic instruction for all living organisms. It determines many traits such as eye color, height, and even whether someone may develop certain disease. Sometimes, problems in DNA called genetic mutation can cause disease. With CRISPR, scientists may be able to correct these mutations directly in the DNA. Interestingly, CRISPR was originally discovered in bacteria. Bacteria use CRISPR as a natural defense system against viruses. When viruses attack bacteria, the bacteria store small pieces of the virus DNA in their genome. Later, if the same virus attacks again, the bacteria use those stored sequences to recognize and cut the virus DNA, protecting themselves. Scientists realized they could adapt this natural system to edit genes in plants, animals, and even human cells. CRISPR mainly uses two key components. First is guide RNA. Guide RNA acts like a GPS or guide, helping the system locate the exact section of DNA that needs to be edited. Second is an enzyme called Cas9 enzyme. The Cas9 enzyme works like molecular scissors. Scientists design a guide RNA that matches the DNA sequence they want to edit. The guide RNA leads the Cas9 enzyme to that specific location in the DNA. Cas9 then cuts the DNA at the targeted spot. Once the DNA is cut, the cell tries to repair it. During this repair process, scientists can remove a gene, repair a mutation, or insert a new gene. Because the system can target DNA very precisely, CRISPR is considered one of the most powerful gene editing tools ever developed. One of the most exciting uses of CRISPR is in medicine. Many diseases are caused by genetic mutations. If scientists can fix those mutations, they may be able to treat or even cure certain genetic disorders. For example, CRISPR is being studied as a treatment for sickle cell disease. Sickle cell disease affects red blood cells and can cause severe pain and serious health complications. Using CRISPR, scientists can modify the patient's stem cell so that they produce healthy red blood cells instead of sickle-shaped ones. CRISPR is also being researched for other diseases such as inherited blindness, certain cancers, and genetic blood disorders. Although many treatments are still in experimental stages, CRISPR could represent a major breakthrough in personalized medicine. CRISPR can also help prevent disease before they spread. One interesting example involves mosquitoes, which transmit dangerous diseases like malaria. Scientists are studying ways to genetically modify mosquitoes so they cannot carry or spread malaria parasites. If successful, this approach could help reduce malaria transmission and potentially save millions of lives worldwide. However, this kind of technology must be carefully controlled because it can also affect ecosystems. CRISPR is also very useful in agriculture. Farmers constantly face challenges such as climate change, pests, and plant disease. With CRISPR, scientists can develop crops that are more resistant to pests, more tolerant to drought, and more nutritious. For example, crops could be modified to grow better in harsh environments and produce higher yields. This could have improved global food security, especially as the world population continues to grow. CRISPR has also become an essential tool for scientific research. By editing specific genes, scientists can study how those genes affect biological processes. This helps researchers better understand how diseases develop, how cells function, and how different genes interact with each other. Because CRISPR is faster, cheaper, and more precise than older gene editing methods, it has greatly accelerated discoveries in biology. Despite its potential, CRISPR also comes with important challenges and ethical concerns. One issue is unintended DNA changes, sometimes called off-target effect. This means CRISPR might accidentally edit the wrong part of the DNA. Another concern is editing human embryos, which could lead to permanent genetic changes passed down to future generations. Scientists and governments are still debating how this technology should be regulated to ensure it is used safely and responsibly. Additionally, the long-term effects of gene editing are still being studied. In conclusion, CRISPR is a revolutionary gene editing technology with the potential to transform medicine, agriculture, and scientific research. It may help us treat genetic disease, improve crops, and better understand life at the molecular level. However, it is also important that scientists continue to study its risk and ensure it is used ethically and responsibly. With careful development, CRISPR could become one of the most powerful tools for solving some of the world's biggest challenges. Because of this groundbreaking discovery, Jennifer Doudna and Emmanuel Charpentier received the Nobel Prize in Chemistry for developing CRISPR as a gene editing tool.